So yes, it's delightful to be here and have the opportunity to talk with you about my favorite scientific subject, G-protein coupled receptors, also known as 7 transmembrane receptors. I'm sure most everybody in this room is aware of the fact that this is the largest and the most versatile and the most ubiquitous of the several families of plasma membrane receptors, anywhere from 800 to 1,000 different members of the family in humans. They regulate a number of our sensory phenomena. I'm looking forward to hearing more about taste receptors a little later in the day from Charles. But probably of greatest significance is the fact that these receptors constitute by far the largest class of drug targets. Hard to get a good number on this. I recently read a review. I'll take the numbers as gospel. I don't know if they are. In the United States anyway, there are 700 FDA-approved drugs targeting GPCRs. They target 135 of the receptors and therefore constitute 35% of all drugs in clinical use in the United States. So this is a big field of study and one of important implications for clinical medicine as it's practiced today. So my lecture, as with several of the ones we've heard already, is going to start off with an historical review. And I really got into this when I was going back over some of the papers, so I don't know how much time I'll have at the end to talk about some of the things we're doing now, but we'll see. We'll play that by ear. But this is a slide taken from Earl Sutherland's Nobel lecture in 1971. Fairly summarizes the state of the field. My own research career began at the NIH in 1968 as a raw recruit with no research experience. So my research career is just 50 years old this year. And virtually that whole career has been spent studying signal transduction and G-protein coupled receptors, even before we knew there was anything such as a G-protein coupled receptor. So here is the concept that Sutherland had. And he first published this in the late 60s. As you can see, he focuses on the adenylate cyclase, the enzyme he discovered, which makes the second messenger cyclic AMP, as the main target of extracellular hormones and drugs and neurotransmitters. Note, there is no independent receptor moiety. There is no such thing as a G protein. His concept was that the concept of how, it was known, quite well known at the time, that a variety of hormones, catecholamines, glucagon, could stimulate this enzyme. His notion was that their site of interaction was somehow directly on the enzyme itself. Here's a slide I'm not going to dwell on. It gives you an oversimplified view of what's happened to that very modest concept that Sutherland had. Much more complexity. We understand a great deal more about it. So the first part of my talk today is going to be about how do we get from that first slide to this. At some point in talking about the receptors, I'll begin to hang the work more on my own contributions, my own laboratory's contributions. But I want to start off talking about the 1970s. This was a time of remarkable ferment. And really, I think it can be fairly said that most, not all, but most of our basic concepts were set down during this period. So this was the decade of the identification of the components and mechanism of hormone-stimulated adenylate cyclase. And I think any such discussion, of course, has to really begin with Martin Rodbell at the NIH. Marty's lab was directly around the corner from where I worked between 1968 and 1970 with Jesse Roth and Ira Paston. And I would say by 1969, Marty was, and this was before his discovery of GTP effects on cyclase, was thinking in these terms. This was a concept that he had developed together with an endocrinologist named Oscar Hector, and they were beginning to talk about this as essentially a three-component system. A discriminator, which we would now call a receptor for hormones, a transducer, the nature of which was completely unknown, and an amplifier, which he perceived as the adenylate cyclase discovered by Sutherland. There were 2 classical papers by Rod Bell. And for the young people in the room who only want to publish in Science and Nature and Cell, as these things go by, I want you to notice where the papers, the founding papers in the field, are published, okay? Count them up. You'll see almost all of them are in JBC, a few in PNAS, 1 or 2 maybe in Science. But JBC, Journal of Biological Chemistry, is where we generally publish this work. Now, so I want to tell you, I'm going to try to spice the talk up with a few personal vignettes, which you may or may not find interesting. But let me at this point share with you a little story about my unintended, minimal contribution to Marty's discovery. So I worked around the corner. This was my very first research experience. I was assigned the task in 1968 of trying to develop a radioligand binding assay for ACTH receptors. I'll skip the part about why ACTH receptors. It was known to activate adenylate cyclase. It was a difficult job, one I was not fully prepared for, but after about 18 months of flailing, I finally succeeded in the task. This was the first scientific paper I ever published. It was in PNAS in early 1970. In late 1969, Marty invited me to present my findings at his lab meeting. This was well before I had published. This was the very first time I had ever presented scientific data outside my own laboratory. I did so. It was not well received. Marty and his main postdoc, Lutz Birnbaumer, who subsequently became a good friend and collaborator, thought this was a foolish way to study hormone receptors, and that the accepted way, as they were doing, was to infer the properties of the receptors by looking at the ability of hormones to stimulate cyclic AMP. So I left, somewhat chagrined, and reported to my bosses that this work was not very well received. About a week later, I go down to the end of the hall to get my every 2-week allotment of a millicurie of 125I to iodinate my ACTH, and I see that Lutz Birnbaumer, who's Marty Rodbell's postdoc, has signed up for the first time for iodine. So I said, Lutz, how come you're signing up for iodine? I mean, you guys don't do anything with it. He says, well, we're going to iodinate some glucagon. I said, how come? He says, we're going to do some binding studies. I said, binding studies? You told me you don't believe in any of that. He says, we don't, but Marty wants to try it. Okay, so back to Marty. They tried it. Within 2 weeks, they had a lovely binding assay going. And then they did a key experiment, which we never did. We had noted, shown in our paper, that the binding kinetics were very slow. But it was known that activation of adenylate cyclase was very fast. And Marty immediately wondered, why the discrepancy? He said, well, initially they used the very same buffers I was using. It was a nondescript tris magnesium buffer. He said, let's do the assay under the same conditions we do adenylate cyclase assays. So they put in all the ingredients, which of course included substrate ATP. Boom, the glucagon binding went down by 90%. So, wow, that was kind of surprising. So what do you do if you're a biochemist and ATP does something? You try other nucleotides, right? So we tried ATP, GTP, CTP, UTP. They all work to some extent or another, but GTP is like 10,000-fold more potent than anything else. Wow, what's that all about? So they said, look, GTP has this profound effect on glucagon binding. Maybe it has some effect on cyclase. Well, it didn't seem to. But then Marty was shrewd enough to realize that it was probably GTP contaminating his ATP. So they enzymatically synthesized some APP-NHP and used that as the substrate. And now all of a sudden, they couldn't get hormone stimulation until they added back GTP. And that's how the discovery of the GTP requirement was made. And that was what was in this paper. Or maybe it was this paper. Yeah. So here was the binding assay, glucagon, and here was the discovery that GTP was absolutely required for hormone stimulation. And that was the key discovery, which really opened up the whole field. At that point, the question became, well, was the GTP acting directly on the adenylate cyclase, or was there a separate component? And several many people worked on that. And of course, Gilman appropriately gets the major share of credit for this. But I just wanted to mention one other person, that's Helmreich, and his postdoc, Thomas Pfeiffer, because they were kind of in lockstep with Gilman's laboratory in isolating a discrete component from membranes with which GTP interacted. The work was not as clean, was not as precise, was not as compelling, but in these two papers, one of them, which I think is probably even a little earlier than Al's, first indicated a separate protein related to adenylate cyclase, which was the nucleotide binding protein. Subsequently, Pfeiffer, in his own lab, was able to do a reconstitution of sorts, but without going into the details, it was not all that compelling. But again, they were right there in terms of trying to identify a GTP binding component. But of course, it was Ross and Gilman, notice the papers in JBC, it was Ross and Gilman who really put the flesh on the bones here. The key to this work was a very interesting variant cell line of the S49 lymphoma cell, which had been isolated, I guess in the late 60s or early 70s by Gordon Tompkins. This cell line was called Cyc-, CYC-, because it had no adenylate cyclase activity. I won't go into how they isolated that. But the key observation that was made by the Ross and Gilman team was that if they took wild-type membranes from wild-type cells, they could heat inactivate the adenylate cyclase. So now you had Cyc- membranes with no adenylate cyclase, wild-type membranes in which there's also no cyclase because you inactivated with heat. You could make extracts of that heat inactivated system, add it to the Cyc-, and now you had very nice cyclase. So there was something that was heat stable that was separate from the adenylate cyclase. They then used that as an assay, the ability to restore hormone responsive adenylate cyclase to Cyc- as an assay to demonstrate the existence and then ultimately to isolate the GTP binding protein, and which today we call GS. Now, enter Zvi Selinger and Dan Cassell. I'm telling the story in a linear fashion, but as any of us who were working at that time know, this was not linear. Things were happening so rapidly in this period from, I would say, 74, 75 through 80, 81, 82, that you opened JBC and PNAS and you held your breath. You just didn't know what was coming. It was all happening at the same time. So the remarkable contribution that we've heard about already and we'll hear more about by Dan and Zvi was the realization that in association with adenylate cyclase activation by hormones, there was activation of a GTPase activity. This was a remarkable biochemical tour de force because, as you can imagine, even if you're not a membrane biochemist, there are a lot of enzymes that can hydrolyze GTP in membranes. And finding assay conditions where you could actually demonstrate the hormone activation of this minuscule GTPase activity was really a remarkable feat. But they did it and showed that there was activation of this GTPase activity by hormones, and immediately made the leap that this was somehow related to adenylate cyclase activation. The key paper, at least in my mind, is this second one on the mechanism of adenylate cyclase activation by cholera toxin, because it had been found by others that, and was known at the time, that cholera toxin, which leads to horrendous diarrhea, does so by permanently activating adenylate cyclase, leading to this tremendous flow of salt and water into the gut. But the mechanism by which it activated adenylate cyclase was not clear. It had been shown by, I think, Gill, as I recall, that it was NAD requiring in an in vitro reaction, and he showed that ADP ribosylation of what turned out to be the G protein, GS alpha subunit, was in fact the mechanism of adenylate cyclase, or was at least closely linked to adenylate cyclase activation. But it still wasn't clear how the two related, and that was the contribution really made in this paper, when they showed very clearly that cholera toxin basically blocked the GTPase activity, which must be the turn-off mechanism, and hence you had persistently activated adenylate cyclase. And so in these two papers was born an idea that has lasted to the present day, the fact that there were these two interlocking cycles of inactive G protein going to active G protein, and that the cycle of GTP activation and hydrolysis was intimately linked to the activation of adenylate cyclase. That could all be blocked by cholera toxins. So now we had part of the story. We had sort of two interlocking cycles, one of GTPase activation and hydrolysis, and one of adenylate cyclase activation, which was then terminated each cycle by the GTP hydrolysis. Now, entirely contemporaneous with this was work coming from the other side, and this is where my laboratory was primarily involved. And again, the groups here in Israel, as just heard, were also working in this area. But the opening salvos in this part of the story had to do with the development of radioligand binding methods. Pretty much contemporaneously, within 2 or 3 months of each other, 3 different laboratories developed assays using radio-labeled antagonists to study the beta-adrenergic receptor. This was one from my own laboratory, but also the one from Daphne Atlas and Alex here in Israel, and contemporaneously, or a few months later, Jerry Auerbach's laboratory at the NIH. In addition, a year or 2 later, my laboratory also developed a very high affinity catecholamine ligand, we call hydroxybenzyl isoproterenol, which allowed us then to tag the receptors both with an antagonist and with an agonist. Subsequently, we went on to develop other ligand bindings assays for alpha adrenergic receptors, etc. Now, once we had radio-labeled ligands in hand, now there was lots that we could do. I want to highlight three observations we made, again, during the exact same period from 75 to 80. First, we wanted to test the idea of Sutherland that the receptive mechanism was in fact a site on the cyclase. It was not. Lee Limbert, a postdoc in the laboratory, was able to show that if we solubilized membranes and chromatographed them on size exclusion columns, we could clearly separate the adenylate cyclase activity from the radioligand binding activity. So the cyclase and the receptor were clearly distinct entities. Next, we made this interesting observation. We took membranes, we labeled the receptors either with our radioligand antagonist or with our very high affinity agonist, solubilized, chromatographed, and lo and behold, the agonist labeled receptor eluded, the antagonist labeled receptor eluded first, then the agonist labeled receptor came out at larger apparent molecular weight. So clearly there was an agonist-induced increase in apparent receptor size. And then collaborating with Gil, whom I told you had developed conditions for NAD-dependent ADP ribosylation of the G protein, we were able to show that this increase in size was due to the formation of a complex between the receptor and the ADP ribosylated substrate, which was the G protein. So here was the first clear physical evidence that there was an RG complex. Now, this jibed very nicely with some functional observations that had been made, again, simultaneously. There was a lot of simultaneity going on here in Gilman's laboratory and my laboratory, both in 1976. And this was of the fact that GTP had effects on agonist binding that it did not have on antagonist binding. Now, we had known since Marty's studies in 1971 that agonists formed a very high affinity binding species, which was then essentially disrupted by GTP. This extended this to the beta receptor, but unlike the glucagon system that he worked with, where he had only an agonist, glucagon, we had all manner of drugs that we could play with. And so the key phenomenology, and this is taken from one of our papers, that we observed is the following. If you look at radioligand competition binding curves, for antagonists, like alpranolol, you got a steep uniphasic Michaelian curve. But for agonists, like isoproterenol, you got a very shallow curve, as shown here, which could be resolved into high and low affinity components. And the addition of GTP completely disrupted the high affinity site. Now you've got only low affinity binding, which again now is uniphasic and Michaelian. And basically, this low affinity matches exactly the low affinity of this hybrid curve. So two affinities. And so you could, using computer-based methods, basically deconstruct these curves and get a high and a low affinity binding component. The interesting observation we made is that if we used a series of beta-adrenergic drugs, which varied in their intrinsic activity, that means their maximum ability to stimulate an effect, like cyclic AMP, setting something like isoproterenol or epinephrine equal to 1, and now you have partial agonists, etc. This ratio of affinities of the low to high correlated beautifully with the efficacy. In other words, to the extent that a high affinity complex could be formed, to that extent was the drug an actual agonist or full agonist. At the time, this finding really got under the skin of a lot of classical pharmacologists, because what we were basically saying was that absent any biological assay, absent any functional assay, we could take an unknown drug and tell you what its efficacy is, simply based on doing this kind of a binding curve. And of course, then with André de Leon in the late 70s, we were able to develop this ternary complex model, which basically postulated that there were two forms of the receptor, a low affinity complex with agonist, which then, by virtue of its combination, forming of a complex between R and something else, ARX, formed the high affinity, okay? And all of these seem to be modulated by nucleotide, which reverted high to low affinity, as I've just shown you. By this point, by the time when we started doing this work in 77 or 78, it really was X. But by the time we published, it was quite clear that X was the guanine nucleotide regulatory protein. And so, to close out this era, this is a model that we published in a review in the early 80s. Nothing original here. There were numerous reviews during this period of time, all of which showed the same model. I'll tell you in a minute why I selected this one from our paper in a second. But basically, it brings all of the work I've just shown you together and shows there were really three interlocking cycles of activity, with the key communicator between the receptor cycle and the cyclase cycle being the G protein cycle. So you have the formation of the high affinity agonist receptor G protein complex, which leads to the loss of GDP from the inactive site. GTP comes on. The GTP liganded G protein then complexes transiently with the cyclase to activate it. GTPase, this goes to inactive, and round we cycle. And so that is the basic mechanism of hormone receptor activation of adenylate cyclase, featuring these two complexes. Now, why did I show you this slide? I wanted to make the point, and again, just a little personal vignette, that in Marty Rodbell's mind, he always referred to this protein as the N protein. Now, Gilman sort of had naming rights because he purified it. He called it G protein. But for several years, we were still calling it N protein because we were collaborating with Lutz Birnbaumer by this time, and he called it N protein. You've already sensed from the introduction there was a lot of competition going during this area. And there were various ideas as to why Gilman called it the G protein. Now, there were some, not me, who thought it was for Gilman. And I can absolutely personally attest to the fact that for a number of years during this period, and somewhere I have a photograph of it. Birnbaumer's license plate was N, not G. That's a true story. Anyway, in the end, it turned out to be the G protein. Okay, moving on to the 80s. This was the era of purification, reconstitution, and cloning of the components of hormone-stimulated adenylate cyclase and discovery of the superfamily of GPCRs. You've heard a little bit about this. My laboratory, while all this mechanistic work was going on, was working very hard to purify the receptors. A daunting job. They were basically trace contaminants of the plasma membrane. We were insanely jealous of people studying the nicotinic cholinergic receptor or rhodopsin, each of which, the nicotinic receptor in the electric fish, electric organ, is about 80% of the membranes. Similarly for rhodopsin and cow retinas. These receptors required several 100,000-fold purification after you found a detergent capable of solubilizing them in an intact form. The key to our success was the development of biospecific affinity chromatography supports, which over a period of a decade we developed for all 4 of the then known adrenergic receptors, beta 1, beta 2, alpha 1, alpha 2, coupling these affinity procedures with a variety of more conventional chromatographic approaches. We were, by the mid-80s, able to purify all 4 of the receptors, 3 of them shown here, to homogeneity. We also developed photoaffinity probes for each of them, and you can see both by protein staining and photoaffinity labeling. Each was a pure species, generally 60 to 65,000 Daltons. They were all glycoproteins. Each one of these species bound radioligands with just the right specificity and stereospecificity that would be expected for the appropriate receptor. So we felt confident we had at least the ligand binding component of the receptor. But of course receptors do 2 things, right? The discriminator of Rodbell, on one side it's binding ligand, on the other side it's talking to the G protein. So it was necessary to show that the same protein was capable of activating a biological response. And here we took a little clue from work of Mickey Schramm. So what Mickey had done is he had shown that he could take 2 cells, each of which contained a receptor-stimulated adenylate cyclase. He was working with Turkey erythrocytes, but then other cells. And using different group-specific reagents, inactivate either the cyclase component or the receptor component, and then fuse them together and show he could reconstitute a hormone-responsive cyclase. So now, but that was just using whole membranes, basically. So now that we had pure receptor in hand, we basically put that receptor into phospholipid vesicles and fused it with a cell which didn't have beta receptors. Now it turned out it was not so easy to find a cell which doesn't have beta receptors. The beta 2 receptor is present in all mammalian cells, and we were isolating it from frog erythrocytes for reasons I won't go into. But it turned out we had discovered somewhat inadvertently that Xenopus laevis, African clawed toad, erythrocytes don't have any beta receptors at all. So that was helpful to know. We took our receptor-containing vesicles, fused it with these cells, which did have prostaglandin receptors, but no beta receptors. So this cell does not generate cyclic AMP in response to beta-adrenergic agonists, but once we had reconstituted it with our pure receptor protein, now it did. Within a year, and working together with Lutz Birnbaumer, who purified the N protein in deference to him, and the late Eva Neer, who purified the cyclase, we were able to reconstitute the entire system in vesicles and show that with only these 3 proteins, pure beta receptor, pure G protein, and pure catalytic cyclase, none of them cloned, by the way. This is before any of these things were cloned. So all of these things had to be purified from endogenous levels of expression. We could reconstitute hormone-responsive cyclase. About a year later, Gilman's lab achieved a similar reconstitution. This proved a number of things. Amongst them, though, was that these 3 proteins and these 3 proteins only were sufficient to establish a hormone-responsive adenylate cyclase. The next step was to clone the genes and cDNAs to understand what the receptors looked like. This was made possible by the fact that we had purified the receptors, microsequencing techniques were advancing. We were able to get several stretches of amino acid sequence. I think we had 5 peptides totaling about 80 residues. And we set out to try to clone the cDNA. This turned out to be difficult. Amongst other problems, we had no experience working with nucleic acids. But fortunately for our group, we had a young cardiology fellow working in the group who was fearless, and that was Brian Kobilka. And after the first nonsensical project I gave him went nowhere, he said, let me have a shot at this. We had been collaborating for some time with a laboratory at Merck, and some more difficult days ahead. But finally, after a lot of trying and some very innovative approaches, such as actually originally cloning the gene, which turned out to be intronless, from genomic DNA before the cDNA, we were successful in cloning the gene and cDNA, first for the beta-2 adrenergic receptor, and then very quickly thereafter for the alpha-2 adrenergic receptor. And of course, in the clone sequence, we could see all the hallmarks of what we take as classical for GPCRs today, the 7 transmembrane domains, sites for N-link glycosylation toward the amino terminus. What came as a bit of a surprise, or more than a bit, as a real surprise at the time, and hard to imagine at this time, is that shown in blue are residues that were actually identical to those in rhodopsin. And of course, rhodopsin was the only other mammalian protein known at the time which seemed to have 7 membrane spans. And so it was the realization that there was a relationship between the beta receptor and rhodopsin that was this real big revelation at the time, and came as a big surprise, despite the fact that we all realized that there were clear functional analogies between the rhodopsin, transducin, a G protein, phosphodiesterase system, cyclic nucleotide, cyclic GMP phosphodiesterase, and the obvious beta receptors. You had sort of a discriminator, a G protein, and a cyclic nucleotide modulatory protein, but still nobody imagined that rhodopsin would have any relation to the beta receptor. So in our initial paper on May 1 of 1986, we speculated that perhaps the whole family of GPCRs, which at that time we would have said was maybe 25 or 30, might look like this. Very quickly, Brian was able to clone the alpha-2, again based on protein sequence that we had gotten after years. John Regan, postdoc, spent 7 or 8 years purifying this alpha-2 receptor from human platelets. And then over the next few years, we would go on to clone a total of 8 adrenergic receptors and a serotonin receptor, and they all looked like this. And so really by the late 80s, we understood that there was a large family of GPCRs. They all shared many, many features. And then the family just expanded dramatically as others got into the game in one fell swoop. Buck and Axel in 1991 published several hundred olfactory receptors, and the family grew. But after our initial work using protein sequence from these purified receptors, almost no GPCR was ever cloned again based on protein sequence. They were all done by homology techniques. So we always felt good in retrospect, and I still do, about that very difficult decade that we spent developing these techniques to purify the receptors, which in a sense served as the Rosetta Stone for getting the larger family. The 80s and into the 90s was really the decade of discovering the mechanism of GPCR desensitization. This phenomenon was well known in the field, shown here for the beta receptor. If you add an agonist, the cyclic AMP goes up, and then within a few minutes declines, even in the continuing presence of the agonist. Here is the same thing for the angiotensin receptor, which is coupled to a different protein, GQ, and which leads to diacylglycerol generation, not cyclic AMP. But it's a very universal phenomenon. The mechanism was completely unknown. And again, a personal vignette. So how did we discover this mechanism of desensitization. Well, about 1980, we had recently developed photoaffinity probes for studying the beta receptors. And whenever we got a new technique, I always wanted to bring it back to the desensitization to see if we get any clues. So I asked one of the postdocs in the lab, Jeff Stadell, to desensitize some cells. And believe it or not, I believe these were actually turkey erythrocytes. And desensitize them by incubating them with agonists, and then photoaffinity label to see if anything was different. And sure enough, the desensitized cells, the receptors, photoaffinity label, migrated more slowly than the non-desensitized cells. And we didn't quite know what to make of that at the time. Remember, this is about 79 or 80. So I went to a Howard Hughes Medical Institute meeting where I showed that slide, and my colleague, then a Hughes investigator himself, Ed Krebs, looks at that. He gets to the microphone. And he says, Bob, he says, I'll bet you that's due to phosphorylation. I said, well, why would phosphorylation change? And we'll take this now as, a given that you phosphorylate membrane proteins, you alter their mobility in SDS gels. At the time, this was only known to the mavens, if you catch my meaning. Ed was such a maven. He said, yeah, I'll bet that's phosphorylation. So I was real excited about that. I called Jeff on the phone. I said, get some inorganic 32PI, label the cells, let's see. In the first experiment, it was clear that the receptors were phosphorylated. And so we knew then that phosphorylation of the receptors was somehow associated with their desensitization. Enter a remarkably talented graduate student named Jeff Benovic. This makes you feel old. In a couple of weeks, Brian and I are going to a retirement party for Jeff, who's just retired from being chair of the Department of Biochemistry at, I always get it wrong, it's Temple, isn't it? Not Jefferson, I always confuse it. At Thomas Jefferson. He's had a great run, but he was an amazing graduate student. And Jeff was able to discover the enzyme, a novel enzyme responsible for phosphorylating the receptor to desensitize it. It was a novel protein kinase. Interestingly, its substrate was only the agonist-occupied receptor. No activity at all against the same. That's how conformationally sensitive this enzyme was. You present it with unoccupied receptor, won't touch it. That was its key feature. He was able to purify it to homogeneity from bovine brain, clone its cDNA. It was the founding member of a new subfamily of kinases, most closely related to the PKA, PKC family. We were aware of contemporaneous work, which had also been going on for a number of years, in the rhodopsin system, where people had shown that in association with its activation, rhodopsin was rapidly phosphorylated by an enzyme being called rhodopsin kinase. We have, in analogy, we're calling our kinase BARK for beta-adrenergic receptor kinase. I got hold of Chris Palczewski, who worked in Paul Hargrave's lab. They were one of the few labs in the world studying that enzyme. Talked him into making one prep for us. We got some sequence, and then we went ahead and we were able to clone the rhodopsin kinase, and sure enough, it was a member of the same gene family. Over the next few years, we, and then Jeff in his own lab, were able to clone a total of 7 members of this family. John Tesmer has basically gotten crystal structures over the last 15 years for most, or perhaps now all of the members of the family. We did the first one with him some 15 years ago. This was for BARK, now known as GRK2. It showed a central kinase catalytic domain flanked by 2 regulatory domains. 2 of the kinases, GRK2, which was the original BARK, and GRK3, interact with the beta-gamma subunits of G proteins. This is a mechanism that helps to translocate them from the cytosol to the plasma membrane. So this was the structure. All of the structures are very similar. Interestingly, of the 7, only 4, GRKs 2, 3, 5, and 6, are universally distributed. So it's a small family, and their range is wide because they can phosphorylate most GPCRs. Now, the discovery of beta-arrestins, which is the second part of this system, Jeff, when he was purifying the bark enzyme, used as his assay for activity purified beta receptor reconstituted in phospholipid vesicles. And he would use two assays. One, he would look at the specific activity as he purified his enzyme for phosphorylation of the receptor. The other, we would then reconstitute G protein and see how much desensitization we got. But the distressing observation was that the enzyme seemed to lose its ability to desensitize this isolated beta receptor as we purified it, which led, as you can imagine, to all kinds of gnashing of teeth. But we correctly, as it turns out, thought perhaps we're losing some necessary cofactor, something else that you need during the purification. Well, while we scratched our heads about that, Hermann Kuhn, this is now 87, published a paper showing that an abundant retinal protein, which to that time was called either 48K protein or S antigen, seemed to somehow work with rhodopsin kinase to deactivate rhodopsin, and he didn't actually, others renamed it now Arrestin, because it seemed to arrest retinal signaling. This sounded like something like what maybe we were losing, something like Arrestin. But of course we knew it couldn't be Arrestin because it was well known that its expression was limited to the retina. So I called Kuhn. I said, can you send me some of this 48K protein? He did, and Jeff, in short order, was able to show that in his reconstituted system, but only at high concentrations, it did restore the ability of bark to desensitize the beta receptor. So, again, fortunately for us, just at that time, everything happening at just the right time, Shinohara at the NIH clones S antigen of 48K. We, speculating that perhaps what we're losing in our system is something which is not only functionally analogous with Arrestin, but perhaps structurally homologous with it, ask him for his clone. We do low stringency cloning, and Martin Losey, then a postdoc in the lab, pulls out a 70% identical protein, which we called beta-arrestin 1, and then a year or two later, another postdoc, Hovated Tramadol, pulls out beta-arrestin 2. And now, with these three arrestins in hand, visual arrestin and the two forms of beta-arrestin, we're able to set up two parallel reconstitution systems, one in which we have rhodopsin phosphorylated by rhodopsin kinase, the other in which we have beta receptor phosphorylated with bark. And now we look at the ability of now the three cloned arrestin species to shut down signaling. And what we find is that for the visual system, arrestin is very potent, the beta arrestins very weak, but for the beta receptor system, the two beta arrestins, are now very potent, and the visual arrestin is very weak. And so we knew at this point that there was tremendous conservation of mechanism between the visual system and the hormonal system. Turns out they're entirely general for all of the GPCRs, but that there was a great deal of specificity between the visual system and the hormonal systems. Today we know there are 4 members of the arrestin family. Unfortunately, there's confusion in the nomenclature in that what I generally have called Barrestin 1 and 2 are also generally referred to as Arrestins 2 and 3. But anyway, Arrestins 1 and X-arrestin are found only in the retina. Now, the 1990s and 2000s, bringing things more into the closer to the present, was an era of discovering the expanded roles of arrestins. So, as I've just shown you, the two-step system that leads to desensitization is shown here. The receptors are activated. Only the activated form of the receptor is recognized by a G protein coupled receptor kinase, or GRK, phosphorylated, usually on multiple sites on the carboxy terminus, leads to Barrestin binding, which we presume by a steric mechanism blocks G protein signaling by occluding the G protein binding site on the receptor. Within 5 years, Mark Carone, one of my early postdocs and a close collaborator, and Jeff Benovic independently both discovered that beta-arrestin it has another role, namely it links the receptor to clathrin AP2. We subsequently found other elements of the clathrin-coated machine to which beta-arrestin could link, and that this is a mechanism, probably the major mechanism for internalization of GPCRs, another very universal process. Then I would say around 2000 we begin to discover in my laboratory that the beta-arrestins are also scaffolds or adapters which can link the Barrestin, link the receptors to signaling cascades, initially SARC, then various MAP kinases, and now a whole host, dozens and dozens of signaling pathways are known to be activated by receptors, not primarily through G proteins, but through Barrestins. So you have two distinct mechanisms of activating many signaling pathways. Classical G protein second messenger signaling, beta-arrestin scaffolding. Sometimes these two work together, sometimes quite separately. And then a discovery we made, I would say about a dozen years ago now, which was perhaps, well, this was already a very shocking discovery. I mean, the proteins were named and had been discovered for stopping signaling, and here we're finding that they actually are a signaling system in its own right. So that was a big surprise. But equally, if not more surprising, was something we discovered, as I say, about a dozen years ago, which was that there were some ligands, drugs, which were able to activate one of these arms of signaling and not the other, or predominantly one and not the other. We call these biased ligands. This was very surprising because you would think if they were, as you know, in the old days, as we thought of receptors existing in two conformations, inactive and active, R and R star, well then all signaling has got to be some function of the amount of R star. So it's hard to imagine how you could have a ligand that could activate some kinds of signaling and not others. It basically requires that there be multiple active conformations, which Brian's work has shown us in spades, and work from my laboratory and other laboratories has brought us to the more modern conception, and you'll hear a lot more from Brian, who's really been the pioneer in all this, that the receptors are just remarkably dynamic, and they exist in multiple conformations, both in the resting or inactive state, or even in the active state. And so what we'd be saying here is that there are some conformations that can lead to all forms of signaling, but perhaps others which lead to primarily Barrestin or G protein signaling. Now, there are also therapeutic implications of the idea of Barrestin signaling. So for example, let's say that beneficial effects of a drug that you want are going through a G protein, and adverse effects were going through beta-arrestin. Okay, so in a case like this, you might want a drug which is G protein biased. That should give you the beneficial effect without the adverse effects. Let me give you a specific example. Take opiates. The beneficial effects to relieve pain are well known to be mediated through activation of G proteins, specifically the G sub I family. But many of the adverse effects, such as constipation and potentially lethal respiratory depression, tolerance, which is really the need for more drug, which is desensitization, seem to be primarily mediated through Barrestin-mediated signaling pathways. So here you might anticipate that a G-biased ligand would be desirable and should give you the desired therapeutic effect with reduced side effects. That turns out to be true in animal models, also in humans, and such drugs are being actively developed at the present time. And I won't go into it, but one can also imagine situations where the opposite is the case. And I think there's a big interest in pharma right now in developing such biased compounds. But recently, I've gotten particularly interested in another form, potential form of drug, which is also a manifestation, as biased signaling is, of the remarkable conformational heterogeneity and plasticity of receptors, and that is so-called allosteric ligands. So, a word of definition, an orthosteric ligand is a ligand, agonist or antagonist, which binds in the orthosteric pocket, defined as that pocket, generally in GPCRs facing the exterior of the cell, into which the normal endogenous ligand binds. That's where adrenaline binds in the adrenergic receptors, where histamine binds in the histamine receptors, serotonin, the serotonin receptors. Allosteric ligands are ligands which bind anywhere else, okay? Now, let me point out that of the 700 or so GPCR drugs that I mentioned before, 698 are orthosteric. There are only 2 allosteric drugs currently approved. So basically, this is a huge, untapped field. Now, allosteric drugs, as I said, could bind anywhere else on the receptor. They could have signaling effects of their own, but more commonly, they either enhance or diminish signaling by the orthosteric ligand. These are referred to as either PAMs, positive allosteric modulators, or NAMs, negative allosteric modulators. Why have I become interested in allosteric ligands over the last few years? Well, there are several reasons. I don't see my own scientific program as being primarily directed at developing any drugs, but there are a number of advantages of allosteric ligands over orthosteric ligands, which I think will make them increasingly valuable in the years ahead. For example, they tend to be more selective. And this is because allosteric binding sites are evolutionarily less conserved. They don't have the same evolutionary pressure to bind whatever it is, adrenaline, histamine, etc. It's particularly so for where there are very closely related, structurally related subtypes of a receptor. There are more divergent sites and actions which can be targeted. Multiple allosteric sites can exist on a particular receptor. Greater on-target safety. Allosteric modulators only affect the activity of receptors when endogenous agonists are present. And so the effects of an allosteric modulator are saturable. It's generally referred to as ceiling effects, which potentially reduces the adverse effects caused by an overdose. That is to say that basically the effect of the drug is limited by the inherent allosteric effect it has on the orthosteric ligand. And perhaps the most important one for some of the work going on in my own laboratory right now, they provide very useful tools for the study of receptors by biophysical techniques. And in particular, I mean, one of our long-range goals is to try to understand what types of conformations might be responsible for example, different forms of bias signaling. But as I showed you, okay, so allosteric modulators are able to freeze or lock receptors into specific conformations by virtue of their cooperative interactions with orthosteric ligands. And I was making the point just a few minutes ago that the receptors are sampling many different conformations, and even when they are occupied by very high affinity, powerful agonists, they're still not locked into any one specific conformation. However, often if an allosteric, a second allosteric modulator is added together with an orthosteric ligand, because of the cooperative interactions between these two species, you can get one particular conformation which is very stable, which you can then study by biophysical techniques. So one of our goals is to develop a whole toolkit of novel allosteric modulators to help us sort out these different conformations, with the hope that along the way we might come up with some novel material which might actually have clinical benefit. So how are we going to get these novel allosteric modulators? Well, we're taking 3 different approaches. We're using nanobody phage display, nanobodies being single variable antibody domains obtained from several species like llamas, for example. We're using very large RNA aptamer libraries, and we're using very large DNA encoded small molecule libraries. So in the interest of time, and because it's the greatest current focus in the lab, I'm going to talk about just the DNA encoded small libraries, small molecule libraries. So these are libraries that are combinatorially synthesized and are huge. They contain as many as 1 billion different small molecules. Consider for a moment that, and this has been explained to me, that you might say, well, how many compounds are known to exist currently? Well, the answer, I'm told, I can't cite a reference for you. I've been told the answer is 50 to 60 million. Okay, so 50 to 60 million compounds. That's not the number of compounds that exist, but that's the number of known compounds that have ever been annotated. So these libraries, when you think of it, having 1 billion different molecular species, these are huge libraries capable, presumably having binders to just about everything. So, and each one is tagged with a little piece of DNA, and in our case about 80 nucleotides, which essentially serves as a barcode. So when you're all done, you know what you got. So the procedure for each of these approaches is to use as bait a purified receptor, GPCR. And we've done our initial proof of principle with the beta-2 adrenergic receptors. And to then incubate that immobilized purified receptor with the library of self-encoding molecules, allow those that are so disposed to bind, wash away those which don't, elute, and continuously recycle following enrichment by qPCR until you reach a stage where the molecules are sufficiently enriched to permit their identification and characterization. One of the nice features of this approach, which is essentially an affinity-based approach rather than using a functional assay, it's extraordinarily high throughput, obviously, but you can stack the deck in favor of what you want. So for example, let's say we want to obtain a negative allosteric modulator. Well, we might either leave the orthosteric site unoccupied to favor inactive conformations, or maybe even put an orthosteric antagonist in here if we have one. Let's say we want PAMs, positive allosteric modulator. We might put a high affinity agonist in here to enrich for populations of active conformations. Let's say we want a biased ligand, something which favors Barrestin signaling or G protein signaling. We might form a complex between the receptor and either a G protein or a beta arrestin. So we have enriching for populations of the receptor which like to bind a particular transducer. I haven't mentioned it, but I'm sure it's obvious to many of you that the transducers, the G proteins and the Barrestins, are themselves allosteric modulators of the receptor. And that's something we've been aware of for 40 years. And in fact, when you look, if you think back to those shallow binding curves that I showed you, what those were reflective of is the allosteric interaction between the G protein, the X in that our model, the G protein and the orthosteric agonist, which is leading to that high affinity site. Okay, so that's the way we get these things. So what happens when you do this? I'm going to give you 2 quick examples. Compound 15. This was pulled out from our libraries using an unoccupied receptor where we expected to get negative allosteric modulators, and we did. The functional characterization shows clearly that it's a negative allosteric modulator. That is to say, if you do an agonist, isoproterenol competition binding curve, in the presence of compound 15, you shift this to lower affinity. If you look at cyclic AMP activation by the agonist, as you get higher and higher concentrations of compound 15, you inhibit signaling. Similarly, if you look at the ability of agonists to promote beta arrestin recruitment to the receptor, that's inhibited in a dose-responsive way. So, compound 15 is the first negative allosteric modulator for the beta receptor. It is, therefore, the first allosteric beta blocker. All known beta blockers, of which there are dozens, and in clinical use, these are all, as with other GPCRs, those are all orthosteric compounds. And so this is an allosteric beta blocker. Okay. To get PAMs, positive allosteric modulators, we occupied the orthosteric binding site with a very powerful, high affinity agonist. And in fact, we pulled out nothing but PAMs. Compound 6 is an example. Now everything's the opposite of what I just showed you. So here's the isoproterenol stimulation curve for cyclic AMP. As you add more and more compound 6, now it goes up to higher affinity and greater maximum. Barrestin recruitment, the same, goes up. And agonist competition binding, it shifts to the left, not to the right. So this is a positive allosteric modulator. So how do these things work? Well, to get at this, we worked together with Brian's laboratory to obtain crystal structures. And Brian and I, over the last, I don't know what, 7 or 8 years now, have been collaborating again. And it's been wonderful. You know, they say eventually you teach your students and then they teach you. And Brian's been trying to teach me some structural biology with modest success. But in any case, we have obtained crystal structures. This is the work of Xiang Liu in Brian's laboratory in Shanghai. And it shows us some very interesting things about where these allosteric modulators bind and how they work. So here's a structure of beta-2 adrenergic receptor with carazolol, a classical high affinity beta-adrenergic orthosteric ligand sitting in the orthosteric pocket. And lo and behold, here's compound 15, far removed, over here on the intracellular surface of the receptor, nestled in this little pocket. These two views of the receptor are as if you were in the cytoplasm looking up at the plasma membrane. Again, you can see compound 15 nestled in its little pocket here. In this representation, where the transmembrane helices are shown as cylinders, you can see that it makes extensive interactions with the inner surfaces of transmembranes 1, 2, 6, 7, and also this non-transmembrane helix 8, which you can see over here. Now, as Brian has shown, for a number of receptors and others as well, when an orthosteric agonist sits down in the orthosteric pocket, information is transmitted across the transmembrane helices such that they move. And this sort of shows you some of that movement. The largest, a number of them are moving there, but the largest movement is known to be transmembrane 6, which moves as much as 14 angstrom out, enlarging the pocket for the G protein or beta-arrestin to fit in. But because of the extensive interactions of this compound with these transmembrane helices, this essentially prevents this motion, restrains this motion, and therefore it's able to act as a negative allosteric modulator. What about the PAM? How's that working? Well, this is a very recent unpublished structure, and you can see that it too binds on the inner surface. So here we have the orthosteric pocket occupied not by an antagonist, but by an agonist, a very high affinity agonist. And here is compound 6, again occupying a site on the intracellular surface. It's in a different place than the site I just showed you. This one, again shown here, the compound nestled in its binding site. This binding site is composed primarily of residues from the inner surfaces of transmembranes 2, 3, and 4, and the intracellular loop 2. This again, of course, is the active structure, as shown here. For comparison, here's the comparable region of the inactive structure, and you can see that the binding region is quite different. It's smaller. If I remove the compound so that you can compare, you can see this pocket is essentially not present here, and this tells us why this is a positive allosteric modulator. Its binding site is effectively not present in the inactive structure. It has to find a receptor in an active conformation to find this binding site. Having once bound, it then helps to stabilize it in this active conformation, thus promoting a receptor activity, obviously not interfering, not overlapping with the G protein binding site. So this is some of the recent stuff we've been doing on allosteric ligands. We hope to use these and others that we're obtaining for our biophysical work. And of course, we're also interested, particularly with other receptors like the angiotensin receptor, in developing novel materials which can serve a dual purpose so that they could work perhaps as drugs that are more specific. There's been a rash of structures, probably a dozen now, or perhaps more, for different GPCRs. And the major lesson we're learning is that the receptors are literally decorated with allosteric sites, beginning on the extracellular surface, all along transmembrane domains, and as I showed you for the two that we happen to get on the intracellular surface. So there's a tremendous landscape of potential territory that can be used for this. Now, in the last minutes of my talk, I'd also like to focus on another project direction that we've been pursuing for the past 5-6 years, and that has to do with trying to understand this only in a sense recently appreciated phenomenon of Barrestin-mediated signaling and Barrestin-biased signaling. So ever since we discovered the Barrestins, as I told you, I guess it's 35 years ago, our sense has been that there were two major forces driving interaction of Barrestin with the receptors. One, the phosphorylation of the receptor tail by a G protein coupled receptor kinase, and then the conformational changes in the core of the receptor driven by agonist occupancy. But again, there was no real physical evidence for that, just speculation. For a number of years now, we've been using a particular reagent to good advantage to gain some insights into what happens when a Barrestin molecule interacts with a receptor and becomes activated. Now it turns out that GPCRs do not all bind to Barrestins equally well. And my colleague Mark Carone, about 20 some odd years ago, pointed out that there are two broad classes of receptors, which he calls A and B, with the class A receptors being like the beta-2 adrenergic receptor or beta-1. They bind Barrestin fairly loosely, relatively low affinity. And then others, presumably due to some particular sequencing of phosphorylation sites on the tail, bind it much more tightly. An example of one which binds very tightly is the V2 vasopressin receptor. And so a number of years ago we began working with a reagent we call the V2-RPP. It is the synthetic 29 amino acid residue peptide comprising the entire C-tail of the vasopressin receptor. It has 8 serines and threonines, and we knew that this is a very good surrogate for the phosphorylated tail of a GPCR because in vitro it binds avidly to beta arrestins and causes conformational changes which appear to mimic those which are activating when it binds to the receptor. Specifically, the phosphopeptide-bound beta-arrestin interacts with much higher affinity with clathrin or with ERK. So we set out a number of years ago to see if we could obtain a structure of the phosphopeptide bound to Barrestin, to see if we could learn something about the mechanisms and structural basis for activation of Barrestins. To that point in time, the only structures of Barrestin were of the inactive molecule. Well, this turned out to be more difficult than we had thought, and we could never get well-diffracting crystals until Arun Shukla in the lab developed an antigen binding fragment, FAB30, which bound specifically only to the phosphopeptide liganded Barrestin very tightly, stabilized it, and we were then able to purify that species. And again, collaboratively with Brian Kobilka's lab a few years back, we're able to get a 2.8 angstrom structure of the phosphopeptide bound to Barrestin-1, stabilized by the Fab. This was work of Ashish Manglik and Andy Cruz in Brian's laboratory at the time. So a word or two about what activation of Barrestin means in structural terms. Here is the structure of inactive Barrestin. It's taken from our paper, but the original structures were done by Paul Sigler's laboratory, I think 17 years ago. And basically it shows that the protein has two domains, the N domain and the C domain, comprised almost entirely of antiparallel beta sheets. Note in green that the C terminus is actually folded back onto the N domain. There are several flexible loops, the finger loop, the middle loop, and the lariat loop. Now, there are two sets of interactions which appear to hold beta-arrestin in an inactive conformation. And these are referred to as the three-element interaction and the polar core. The three-element interaction consists of these three elements: beta strand 1, this alpha helix, and the Barrestin, in this case 1 C-terminus. And the polar core consists of 5 polar residues, 3 aspartates and 2 arginines at the core of the molecule. In both cases, the C-tail residues, as you can see, are interacting with these other residues on the end domain. So what were the key findings from the structure? First of all, the phosphopeptide binds to the end domain at a location very similar to the Barrestin C-terminus in the inactive structure. If you look at this here in light blue is the V2 peptide is in green, and you can see how closely aligned that is with the normal disposition of the C-tail shown in light blue. So this displacement of the C-terminus. This, of course, disrupts the two key sets of interactions, which I told you stabilize the inactive beta-arrestin, the three-element interaction and the polar core. Now, subsequent to phosphopeptide binding, the N and C domains of Barrestin undergo a substantial twist with a 20-degree rotation around the central axis. You can see this here. In gold is the active structure, in blue the inactive structure. And one might suppose that this rotation, amongst other things, might expose sites on the molecule for interaction with the numerous endocytic and signaling partners that it has the capability of binding. Another interesting finding was that phosphopeptide binding also occludes the inactive conformation of the finger loop, which has been shown to be important for discrimination between active and inactive GPCRs, thus stabilizing this loop in an extended conformation, which may facilitate contact with the receptor core. This shows the extended finger loop, and of course that immediately raises the question, is that finger loop now being able to insert into the core of the receptor? And so, of course, at this point we wanted to get some more information about the interaction of active beta-arrestin with an actual receptor, not just this phosphopeptide. This required some gymnastics and developing a novel system. What we eventually wound up doing is we created a chimeric receptor in which the tail of the vasopressin receptor is genetically fused to the core of the beta receptor. We call this the beta-2 V2 receptor. This conveys on the beta-2 receptor the very tight Barrestin binding inherent in the vasopressin receptor. The beta-2 V2 is expressed in SF9 cells together with Barrestin 1 and a GRK2, which is palmitoylated to keep it on the membrane. Stimulate with agonist leads to phosphorylation of the receptors, formation of the complex. Okay. Now the complex again was unstable unless we added Fab30 to the lysates, which we did. Now we have a stable beta-2 V2 agonist, beta-2 V2 Barrestin Fab30 complex. We're able to purify it. And together with Brian and Yurgo Skiniotis' laboratory, a variety of techniques have been used to sort of study the properties of this complex. Most particularly, Yurgo has used negative stain EM, but we've also done hydrogen deuterium exchange, lysine cross-linking, disulfide cross-linking. You can see that it's a relatively homogeneous, monodisperse population of particles. And Yurgo's lab, when he was at the University of Michigan, now Brian has lured him to Stanford, used single particle classification and averaging to get at its underlying architecture. And here's what he found. So 2/3 of those particles look like this. Here's a little cartoon to help you get oriented. The ovoid density is the beta-2 V2. This is a T4 lysozyme domain, which is engrafted genetically on the amino terminus. A little trick developed in Brian's laboratory. It helps with crystallogenesis of the receptors in crystallography studies. Here it does serve as a very useful orienting feature in the microscopy. Here's the Barrestin which appears to hang by a single point attachment, presumably from that phosphorylated V2 tail, and here is the Fab30. So we envisaged or speculated this might represent the very first interaction of Barrestin with the receptor. I said that's about 2/3 of the particles. 1/3 look like this, and they feature a tighter, more intimate interaction of Barrestin with the core of the receptor shown here, and we again speculated that this might be a secondary interaction whereby the phosphopeptide activated Barrestin binds to the core of the receptor. So 2/3 and 1/3. Using an on-column glutaraldehyde cross-linking technique, we were able to convert all of these to these. So now there was a homogeneous population of these cross-link complexes in this what we call core conformation. Jurgo was able to use those to make a very low-resolution 3D reconstruction shown here. In the EM envelope, you can see densities corresponding to each of the major components, this globular species being the receptor surrounded by the detergent micelle, that T4 lysozyme domain, the beta-arrestin, and the Fab. Using a variety of distance constraints that we had from the cross-linking that I mentioned to you, he was then able to model into this envelope Brian's previously published T4L beta-2 receptor structure and the beta-arrestin 1 phosphopeptide Fab30 structure, which I just showed you. The only thing I want to point out is that the low-resolution reconstruction confirms extensive interactions of the phosphopeptide tail with residues in the end domain, and also a deep penetration of the finger loop into the core of the receptor. So what these early structural studies do seem to confirm is that, as we had always thought, there do seem to be two interactions, one with the phosphopeptide tail, which appears perhaps to be the initial interaction, and then the other with the core of the receptor. We call these the hanging and the core conformations. Of interest, this clearly would be a very satisfying arrangement to explain desensitization. And this is sort of, I guess, what we had been picturing low these last 35 years, namely a way in which the Barrestin would sterically block G-protein interaction. But it does leave hanging, if you will, whether this hanging conformation is simply a way station, an intermediate on the way to the core, or whether it might actually be doing anything of its own in its own right. To get at that, we created a mutant form of the Barrestin in which we deleted the entire 15 residue finger loop. And one would envisage that that can no longer form the core, but can only do the hanging conformation. We used negative stain EM to confirm that this mutant could form only the hanging and not the core conformation. And then I'm not going to go through the data because it's getting late, but we were able to demonstrate and published about a year ago that this hanging conformation, using the Barrestin minus the finger loop, can form the core, was able to, was quite active, not fully active, probably about 75–80% active in mediating endocytosis, and also active in mediating the two forms of signaling we looked at, ERK and SARC. Now, that doesn't say it can do all signaling. I mean, there are dozens and dozens of pathways, but at least some signaling and some and endocytosis can be mediated without the core interaction. But what could not be mediated without core interaction is desensitization. So endocytosis, some forms of signaling seem to be mediated by this conformation, or at least they can be, but the core conformation reasonably seems to require the core conformation. But recently, we've discovered another potential role for the hanging conformation. And this has to do with a new paradigm for understanding cell signaling. not one that we discovered. It's been discovered by several groups working with the parathyroid hormone receptor, the TSH receptor, and to a lesser extent the beta-2 receptor. And this new paradigm is sort of depicted here. So in the classic paradigm that we've all understood for decades, agonists activate receptors, they activate the G proteins, and we get, for example, cyclic AMP generation. This is followed by GRK-mediated phosphorylation of the active conformation. Barrestin binds, desensitizes, leads to endocytosis. The receptors then go into endosomes, and depending on whether they're type A, loose binding, or tight binding, if they're loose binding, the Barrestin falls off immediately, and the receptor goes in alone. If they're tight binding, like the V2 vasopressin, they are resident as a receptor Barrestin complex in endosomes for prolonged periods. Some may recycle, but many of these get diverted to lysosomes and are degraded. Okay, but, so here shows the recycling. But the new paradigm is that it's now been shown for a growing list of receptors, especially those ones that tightly bind Barrestin, that they can continue to signal, for example, to cyclic AMP from within the cell. Now, that presented a real conundrum, which we haven't understood, because on the one hand, it seems to require a persistent receptor Barrestin complex to remain resident in endosomes. On the other hand, if it's cyclic AMP is being generated, this receptor presumably is able to couple to G protein. And since the two can't seem to bind together, how could that be happening? Well, as we pondered these two different conformational arrangements of receptor and Barrestin that I've told you about, it did occur to us that in the hanging conformation, almost the entire internal surface of the receptor remains available. And so we speculated that perhaps this hanging conformation could, while still bound to Barrestin via its tail, further complex with heterotrimeric G protein to form this species, which we call the megaplex. And so we set out to see if we could develop evidence for the formation of such a complex in cells. And again, we published that last year. I won't go into it, but we showed extensive evidence, including things like confocal microscopy, Brett studies showing agonist stimulated enhancement of proximity between G alpha or G beta gamma with Barrestins, other such evidence that such a complex is in fact forming in cells. And then we were able to form such a complex by interacting purified heterotrimeric G with this complex. Again, Yurgo Skiniotis had a look at this by negative stain EM. You can see the various components, the receptor with the T4L cap, if you will, beta arrestin in hanging conformation, the Fab, and here on the inner surface, the G protein. Interestingly, if you look at these two negative stain images, this is essentially like the ones I've been showing you for the T4L beta receptor Barrestin 1 Fab 30. This is a previously published image from Brian Kobilka and Yurgo Skiniotis showing the beta receptor and GS complex. And just for fun, if you superimpose those, you can see that it looks very similar to the complex of all of these that you get together. Note that the G protein fills the void on the inner surface of the receptor, and we have shown biochemically that the G protein in this complex is fully capable of being activated by the beta receptor, despite the presence of Barrestin hanging off the tail. Most recently, an MD PhD student in my laboratory, Anthony Nguyen, has been working on a cryo-EM structure. Still a work in progress, but by getting close, he's down to about 4.3 angstrom resolution. This is, of course, the receptor, the Barrestin and the Fab 30. To further stabilize this complex, in order that we could study it, we, in addition to the Fab 30, we had to use two additional immunological reagents, an antibody 32 to the phosphopeptide Barrestin complex, and another nanobody, here it is, 35, developed in Brian's laboratory to the G protein. It's easier to look at this multi-component complex if we take the antibody reagents out, which I've done here. So you can see in the structure that because of the flexibility, very poor resolution of the phosphopeptide tail, but the tail does in fact appear to represent the only point of interaction of the receptor with the Barrestin. Here is the G protein and its various subunits. So clearly the receptor is simultaneously bound to both effectors, both the G protein and the beta arrestin. So we wondered, of course, does the binding of either transducer perturb the binding of the other? And to a surprising extent, the answer appears to be no. So here is the structure of the receptor part of this complex, superimposed on either the inactive receptor or the active receptor, both structures that Brian's lab had published in previous years. This shows the 14 angstrom rotation of TM6 in the megaplex, which matches what was seen in previous active structures of the beta-2, and you can see does not superimpose at all on the inactive structure. But when superimposed on the active receptor, this 14 angstrom rotation and the other rotations of the of these helices match up exactly. So it appears that the receptor conformation is close to identical to that which is seen in the receptor G protein complex. What about the Barrestin? Well, you can't see it well here, but the Barrestin again is in its active conformation. There is this 20-degree rotation, for example. In red is the phosphopeptide. Now, remember, this phosphorylation was carried out in cells, as I explained, unlike the isolated tail, which has 8 phosphoserines. We can only resolve 6 of those, whether the other 2 are just not resolved or whether in the incellular phosphorylation we've not gotten them is not clear. And this just shows the phosphopeptide in red and some of the arginines and lysines on the end domain with which it's interacting. I would point out that in this cartoon, the V2 tail phosphopeptide shown in red. So these are the phosphates and how they line up in the cryo-EM structure, where the V2 tail is part of the receptor. And in yellow is the phosphopeptide from the structure I showed you of the isolated phosphopeptide with Barrestin. You can see they're superimposable. So again, these features suggest that the interaction of the Barrestin in the hanging conformation in this megaplex is similar to what we had previously observed in vitro with the isolated phosphopeptide. And so I'll conclude this by just saying that this last point is that there appear to be these 2 distinct conformational arrangements, presumably many more, but at least these 2 arrangements where the interaction with the phosphorylated tail of a receptor is sufficient to mediate endocytosis and some forms of signaling, but now it looks like there may be another role for that conformational arrangement, namely to further complex with G proteins inside the cell to mediate endosomal signaling. But the core conformation is necessary for desensitization. So I will conclude at that point. This gives you a sense of some of my current interests in the laboratory at the moment, and hopefully a better understanding of the way our field evolved over the past 45 or 50 years. Some acknowledgments for people in the laboratory, both recent graduates, but primarily people who are still there. The Skiniotis lab, who did all the electron microscopy. Brian has been a faithful collaborator and teacher on a number of all of these structures. Shang Yu Liu in Beijing. What did I say, Shanghai before? Close enough, right? And New Evolution and Lexicon are two commercial firms from whom we have obtained the small molecule libraries. Thank you.